Chapter 4. The Origin of Multicellularity. Cells learn to cooperate. 4.1 The Bargain. Somewhere in your body, right now, a cell is dying on purpose. It is not damaged. It is not sick. It has simply received a signal, a chemical instruction, and in response, it is dismantling itself. It shreds its own DNA. It folds up its membranes into tidy packets. It shrinks, it blebs, it breaks apart into neat parcels that neighboring cells will collect and recycle. The whole business takes a few hours and makes no mess. Billions of your cells will do this today. They will do it without complaint, without resistance, and without ever passing on a single one of their genes. Now hold that against everything you have learned in the first 3 chapters of this book. Life is a process that copies itself. That is nearly the whole definition. For 3 billion years, from the first replicating molecules in the vents through the last of the great prokaryotic empires, the rule was unbroken and utterly simple. The things that copy themselves best become common, and the things that don't. And here is a cell, at the peak of health, choosing not to. This chapter is about how that became possible, how life talked itself out of its oldest rule. Because your body is not really you, not in the way it feels. Your body is around 30 trillion cells, and every one of them descends from a lineage that spent billions of years being ruthlessly, magnificently selfish. Almost every one of them has now surrendered its own reproduction forever. Your skin cells will die as skin cells. Your neurons will die as neurons. The entire staggering apparatus, heart, lungs, bones, eyes, brain, this specific mind reading this specific sentence, is a support system, built by cells that will get nothing out of it. For a tiny handful of reproductive cells hidden in the gonads. 30 trillion cells have agreed to die so that a few might reproduce. How on earth did evolution ever arrange that? 4. Point to what counts as a body. First, some care with words. Cells stick together all the time. Not every clump is a body. Aggregation is a crowd. Bacteria in a biofilm dental plaque, say, pile together and gain real benefits from the crowd. But each cell is a full independent organism, free to leave. Nobody has given anything up. This is a group of individuals, not an individual. Colonial organization is a step further. Cells divide and stay stuck to one another. They cooperate a bit, but pull one loose and it will very likely survive on its own, divide and start a new colony. Membership is convenient, not compulsory. True multicellularity is different in kind, and the difference is this. The cells cannot go home again. They are permanently committed. Separate them and they die, most of them anyway. And crucially, they are not all doing the same job. They have specialized. They have divided the labor. And of all the possible divisions of labor, one is the true frontier, the line on the far side of which life is genuinely, irreversibly different. Germ and soma. Germ cells make the next generation. Somatic cells do everything else. They eat, they move, they build, they defend, they think, and then they die, taking their genes with them into oblivion. The moment a lineage of cells accepts that division, something new exists in the universe, an organism, a thing that is more than the cells composing it, with a life of its own that they will spend themselves to sustain. 4.3 The cheater's advantage. Here is why this should not work. Imagine a young multicellular organism, a modest ball of a few hundred cooperating cells. Most of them are sterile workers. A few are reproductive. Now imagine that one worker cell suffers a mutation, and the mutation says, ignore the rules, divide. Ask yourself who wins. Not the organism is now carrying a growing lump of freeloaders that consume its resources and contribute nothing. But within the body, the rebel is winning enormously. Every other cell in that body is producing 0 descendants. The rebel is producing a great many. In the local competition, the one that matters moment to moment, cheating is not merely a decent strategy. It is an unbeatable one. Selection does not care that the rebel will eventually kill its host in itself. Selection has no foresight whatsoever. It sees only what replicates faster now. You already know the name of this problem because it will kill roughly 1 in 5 people reading this page. Cancer is not a disease that multicellular life happens to catch. Cancer is multicellularity's original problem, resurfacing. It is a cell of your body doing exactly what every one of its ancestors did for 3 billion years, dividing whenever it can, taking what it needs, obeying no one, and it is a reversion. A return to an older and simpler way of being alive. Every tumor is a cell lineage that has, in a sense, remembered. This tells you something important about your own body, and it is not comforting. You are not a machine. You are a truce. And a truce is a thing that must be actively maintained every hour in every cell for as long as you live. So the question sharpens. How do you get a truce to hold when defecting always pays? 4.4, the answer is family. The answer is one of the most elegant ideas in all of biology, and it comes down to a single word: relatedness. Ask again what a selfish cell is selfish for. Not for itself, a cell has no self to speak of. Selection acts on genes, and a cell's genes don't care which particular cell carries them into the future. They care only that they get there. Now suppose every cell in a body is a clone, every one descended from a single founding cell, every one carrying an identical genome. A skin cell that dies for you loses nothing. Its genes are already in the germ cells. Every gene it holds is present, atom for atom, and the sperm are the egg that it is helping to build. From the gene's point of view, it makes not the slightest difference whether it reaches the next generation via this cell or that one. Sacrifice, when the beneficiary is your identical twin, is not sacrifice at all. It's just the long way around. This is the deep logic. Genetic identity dissolves the conflict of interest. When every cell in the body has exactly the same stake in the outcome, there is nothing to fight over. Cooperation stops being altruism and becomes simple self-interest, viewed from one level up. And that is why the shape of your development is not an accident. You did not begin as a crowd. You began as one cell, and everything you are grew out of it by division without separation. Every body on Earth built that way is, at bottom, a family so close that it has no members. 4.5, it happened over and over. For a transition this profound, you'd expect it to be vanishingly rare, a once-in-a-planet fluke, like the swallowing that made the eukaryotic cell. It isn't. Multicellularity evolved independently dozens of times. Animals did it once, somewhere between 650 and 800 million years ago. Land plants and their green algal relatives did it separately. Fungi did it more than once within the fungi alone. Red algae did it, and their fossils are the oldest good evidence we have. Brown algae did it astonishingly late. The kelp forests, those great swaying underwater cathedrals, are a recent invention, on the order of 200 million years old, younger than the first dinosaurs. Slime molds did it in their own bizarre way. And that's before you count the many smaller, obscure lineages that made bodies and never made history. Even prokaryotes flirt with it. Some cyanobacteria grow as filaments in which certain cells stop photosynthesizing and specialize in nitrogen fixation for the benefit of their neighbors, a genuine division of labor in a bacterium. And myxobacteria, when the food runs out, do something that ought to be impossible for a bacterium. They stream together by the hundreds of thousands, pile up, and construct a fruiting body, a tower, from which spores are launched. The bacteria that form the stalk are not going anywhere. They built the tower and they will die in it. So the honest summary is this: making a body is not the hard part. Life invents bodies casually whenever the conditions favor it. What is rare, what took a billion years, and in animals happened exactly once, is making a body complicated. Tissues, organs, a nervous system, a shape worth having. Multicellularity is common. Complexity is not. 4.62 roads. There are exactly 2 ways to build a body, and the difference between them decides almost everything about what the body can become. Stay together. One cell divides, and the daughters do not separate. They divide again, still they hold on. Over and over, from one founder, and every cell in the resulting body is a clone of every other. This is how you were made, from a single fertilized egg through some 40-odd rounds of division, without any cell ever leaving. It's how every animal, every plant, every mushroom is made. Come together. Independent cells, living separate lives, converge and assemble into a collective. The great exemplar of the second road is a creature so strange it deserves its own paragraph. Dictyostelium discoideum is a slime mold, and for most of its life it isn't a slime mold at all. It's a population of solitary amoebae, crawling through the soil, eating bacteria, dividing, ignoring each other completely. Then the food runs out. And when it does, the starving amoebae begin to release a chemical signal, and to move toward the signal, and the signal amplifies, and across the soil, tens of thousands of individual amoebae converge from all directions in beautiful spiraling streams, flowing toward a common center like rivers running backward. They pile into a mound, and the mound stands up and topples over and becomes a slug, a genuine multicellular organism, a few millimeters long with a front and a back that crawls purposefully toward warmth and light. It has no brain. It is made of what were an hour ago. Perfect strangers. When the slug finds a good spot, it stops and performs its final act. It lifts itself. About 20% of the cells sacrifice themselves to build a rigid stalk. They secrete cellulose walls, stiffen, and die. And they die lifting, hoisting the remaining 80% up into the air, where those cells become spores, ready to be carried away on the leg of a passing insect to somewhere with food. 20,000 amoebae go into a slug. 4,000 of them become a dead stalk, holding 16,000 up toward the sky. And now you can see, in a single organism, exactly why the Stay Together road is the one that led to us. Because those amoebae are not clones. They came from all over. They're related, some of them, but not identical, and so the logic of section 4.4 fails. A cell that becomes stalk is dying for someone else's genes, which means there is a real, permanent, ineradicable incentive to cheat. And cheaters exist. Some strains of Dictyostelium have been caught doing it, sliding preferentially into the spore fraction, letting other lineages hold them up. The whole society is one long, unresolved negotiation, and it shows. Aggregative multicellularity has never produced anything large or lasting. It stays small and simple and part-time. Come together builds a truce among strangers, and a truce among strangers is always fragile. Stay together builds a body out of a family, and a family can afford to build something that takes 1 billion years. 4.7, the toolkit was already there. You would think that building a body from scratch would require a great pile of new inventions. It didn't. And this is the pattern you should be watching for by now, because it's the third time this book has shown it to you. The parts were already lying around. To build a body, you must solve 3 problems. Stick together. The animal solution is a family of proteins called cadherins, which reach out from a cell surface and clasp identical cadherins on the cell next door, a molecular handshake held closed by calcium. Different cadherins prefer different partners, so cells can sort themselves by type. Mix 2 kinds of cells with 2 kinds of cadherin in a dish comma, and they will spontaneously separate and recluster with their own, which is very nearly the whole trick of building an embryo. Talk to each other. Animal cells punch gap junctions through their shared walls. Open channels, cytoplasm to cytoplasm, through which ions and small molecules flow freely. Neighboring cells become chemically almost one. Plants, arriving at multicellularity entirely separately, faced the same problem, and solved it with plasmodesmata, tunnels bored through the cell wall. Different proteins, different architecture, same idea. Life keeps finding the same answers. Build a scaffold. Cells secrete an extracellular matrix, a mesh of proteins, most importantly collagen, in which they live embedded, like bricks and mortar. It gives shape and strength, provides rows for migrating cells, and carries signals of its own. Collagen is now the most abundant protein in your body. It's your skin, your tendons, your cartilage, the scaffolding of your bones. And to coordinate all of it, cells use a small handful of signaling systems, WNT, Notch, and a few others, which appear again and again throughout development, deciding which cells become what. Now, the punchline? Nearly all of it predates animals. Cadherins are found in single-celled organisms that are not animals and never were. So are the core components of the great signaling pathways. So are pieces of the machinery of programmed cell death. The molecular toolkit that builds your body was invented by solitary cells, for their own private purposes, catching prey, sticking to surfaces, sensing the water, responding to their own internal damage. Evolution did not design a body. It rummaged. It found a set of tools built for other jobs and pressed them into new service, and the body was assembled out of borrowed parts. You saw this with the Asgard archaea in Chapter 3, a cell already carrying the eukaryotic toolkit before there were eukaryotes. It is one of the deepest patterns in the history of life, and it will keep recurring in every chapter that follows. The toolkit precedes the function. Evolution is not an inventor. It is a scavenger of genius. 4.8 A living staircase. Sometimes evolution leaves the intermediate steps lying around where you can look at them. In ponds and puddles all over the world lives a family of green algae, all closely related, that between them display very nearly every stage in the transition to multicellularity. They can be grown side by side in the same lab on the same afternoon. At the bottom of the staircase is Chlamydomonas, a single cell with 2 flagella swimming alone. Wholly independent, wholly ordinary. Then gonium, a flat plate of 4 to 16 cells, stuck together, beating their flagella in loose coordination. A raft. Not much more than that. Then Pandorina and Eudorina, balls of cells now better coordinated. With a definite front and back, the colony swims as a unit toward the light. And then Volvox. If you have never seen one, find a picture. A Volvox colony is a hollow, transparent, glittering sphere, up to 50,000 cells, arranged in a single layer across its surface, every one of them beating a pair of flagella. The whole globe rolls slowly through the water, turning as it goes. Inside it, floating free, are its daughter spheres. Perfect small worlds within the world, waiting to be born. But here is what matters, and it is not the beauty. Of those 50,000 cells, the overwhelming majority are somatic. They swim. They photosynthesize. They keep the sphere alive and moving toward the light. And they cannot reproduce, not slowed down, not discouraged, but permanently and physically incapable. They will divide never again. When the daughter colonies are released, the parent sphere breaks apart and all of them die. A few dozen cells, tucked inside, larger, flagella-less, doing nothing but sitting there, are the germline. They are the only cells in the entire organism with a future. 50,000 cells row a boat toward the sun, so that 30 of them can have children. And then the 50,000 die. This is not a metaphor for our condition. It is precisely our condition in an organism you can hold in a drop of pond water, a body that has struck the same bargain your body has, and can be watched making it. The Volvocene algae matter because they demolish the objection. Nobody has to explain how life leapt from a solitary cell to a differentiated body in one improbable bound, because the leap can be walked. Single cell, small raft, larger ball, coordinated sphere, sphere with a germline. Every step is small. Every step works. Every step is alive in a pond somewhere today. 4.9 are cousins, the collar cells, now animals. Who was the last solitary cell we came from? The best living guide is a creature called a choanoflagellate, a single-celled eukaryote, a tiny oval cell with a whipping flagellum, surrounded by a delicate collar of fine tentacles. The flagellum beats, water flows through the collar, and bacteria are trapped and eaten. They are common in the sea and in fresh water. And they are the closest living relatives of animals. Of all animals. Of every animal that has ever existed. 2 things about them will stop you. The first, some choanoflagellates, in response to certain chemical cues in their environment, stop living alone. They divide and stay together, forming a hollow rosette of cells, an assembly that looks unnervingly like the very earliest stage of an animal embryo. They are not permanently multicellular. They can do it and undo it. It is multicellularity as a facultative option. Something you switch on when conditions call for it. The second, sequence a choanoflagellate genome, and you find cadherins. You find pieces of the animal signaling pathways. You find much of the machinery we spent the 20th century assuming was the unique inheritance of animals, sitting in a solitary cell that scoops up bacteria in a pond. Now put a sponge next to it. A sponge is an animal. It's on the deepest branch of the animal tree. It is about as close to the base of our own family as a living thing can get. It has no nerves, no muscles, no brain, no organs, no mouth, no front or back. It sits on a rock and pumps water through itself. And the cells doing the pumping, lining the chambers inside its body, beating their flagella, catching bacteria in delicate collars, are called conocytes. They look almost exactly like choanoflagellates. The sponge is, in a rough but powerful sense, a colony of collar cells that learn to live together, with other kinds of cells added around them. Amoebocytes that crawl through the sponge's matrix, carrying food, building the skeleton, doing a kind of primitive immune work. It has cell types. It has division of labor. It has a germ line. It is genuinely an animal, and there's a famous, almost unbelievable demonstration of how loose its integration still is. Push a living sponge through a fine mesh, so that it is reduced to a soup of separated cells. A smear, an organism destroyed. Leave the soup in seawater. The cells crawl toward each other. They find each other. They sort themselves out by type. And over hours and days, they reassemble into a working sponge. Try that with a mouse. The sponge sits precisely where this chapter needs it to. Past the colony, short of the body. Cooperating, differentiated, and still just barely able to be pulled apart and put back together. 4.10 The police. Relatedness explains why cooperation can work. It does not guarantee that it will. The reason is mutation. Your cells may have started out as clones of the fertilized egg, but every division introduces errors, and after trillions of divisions your body is a mosaic, not one genome, but a haze of slightly different genomes, all descended from the original. Somewhere in that haze, sooner or later, a lineage will acquire a mutation that makes it divide when it shouldn't. The truce, unpoliced, always breaks eventually. So the body polices. Apoptosis is the mechanism from the first page of this chapter. Programmed cell death. The clean and orderly self-destruction of a cell that has been told to go, or that has decided on its own that something is wrong. Cells run continuous internal checks, and if the damage is bad enough, or if the surrounding tissue withholds the survival signals a cell needs to keep living, the cell dismantles itself. Note that inversion carefully. In your body, the default state of a cell is death. Your cells are not staying alive because they want to. They are staying alive because their neighbors are constantly giving them permission. Checkpoints are the second layer, molecular inspectors that halt the cell cycle if DNA is damaged, if chromosomes are misaligned, if anything looks wrong. Fix it, or don't divide. If it can't be fixed, apoptosis. And the germ-soma divide is itself the deepest defense of all, by declaring, very early in development, that the vast majority of cells will never reproduce, sealing off the germ line. Sometimes within days of conception, the body removes most of the prize. A somatic cell, that rebels and multiplies wildly, can build a large population of its descendants, and it does not matter, evolutionarily speaking, because none of them are going anywhere. When the organism dies, they die with it. Cheating has been made pointless. Made pointless, but not made impossible. This is the crucial and slightly bleak point. The defenses are excellent. You fought off dozens of incipient rebellions before breakfast. But they are statistical, not absolute. Given enough cells, enough divisions, enough decades, some lineage will eventually get past every checkpoint, disable its own apoptosis and defect, which is why cancer is not a design flaw in an otherwise sound machine. It is the price of admission. It is what you pay to be a body at all, the permanent, structural cost of the bargain struck in the Precambrian, still coming due in you. 4.11 What the rocks show. The rocks tell the story badly, because soft things rarely fossilize, and small soft things almost never do. The earliest candidate is genuinely startling, the French-civilian biota of the Bon, centimeter scale, structures preserved in rock about 2.1 billion years old, vastly older than anything else in this chapter. Some researchers see organized, coordinated, multicellular organisms. Others see mineral formations, or microbial mats, that merely look organized. The argument continues, and it would be dishonest to tell you it's settled. The oldest fossil that nearly everyone accepts is a red alga called Bangiomorpha pubescens, from the Canadian Arctic, roughly 1 billion years old, and it is a lovely thing to have found, because it doesn't just show cells in a row. It shows differentiation, a specialized holdfast cell at the base, anchoring the filament to the rock, and cells that appear to be specialized for reproduction. Division of labor, plainly visible in stone, 1 billion years ago. Bangiomorpha has one more secret worth mentioning. It appears to be the oldest known organism that reproduced sexually, with 2 distinguishable types of reproductive cell. The oldest complex body, and the oldest sex, in the same fossil, in the same rock. Then a long silence, and then, at last, the Ediacarans, from around 571 million years ago, until the dawn of the Cambrian. Large, soft-bodied, strange. Dickinsonia, a flat quilted oval up to 1 meter across that looks like nothing alive today. Kimberella, which may have been an early mollusk and appears to have left scratch marks where it scraped food off the seafloor. These are the first indisputable, sizable, complex animals, and they are the last thing this book will show you before everything changes. But there's a gap, and it's a real one. Molecular clocks, which estimate when lineages split by counting up genetic differences and dividing by an assumed rate of change, put the origin of animals at something like 650 to 800 million years ago. The fossils don't show up until much later. Is the clock wrong? Or is the record empty? Probably the record. The first animals were, in all likelihood, tiny, soft, and utterly unpreservable. The kind of thing that leaves no trace at all unless the conditions are perfect, and the conditions almost never are. The first 100 million years of animal life may simply be gone. Unrecorded, the way most of history is gone. Absence of evidence is not evidence of absence, a phrase paleontologists have to repeat to themselves rather often. 4.12 The threshold. Take stock of what has been built. Cells that stick together. Cells that talk to each other. A scaffold to live in. Signaling systems that tell cells what to become. A clonal body, in which every cell shares the same genetic interest. A germ line, sealed away and protected. A somatic body, sterile, committed, and disposable. And a police force to hold the whole arrangement together against the constant temptation to defect. Life has thoroughly, permanently crossed the line from cell to organism. The bargain has been struck, and the truce is holding. But look honestly at what has actually been made with all this machinery. A sponge, a hollow green sphere, a filament of algae with a hole fast at one end. Bodies, yes, but simple ones. A sponge has perhaps a handful of cell types, and no tissues at all, no organs, nothing that coordinates, nothing that decides. The whole grand edifice of cooperation has been erected, and it has produced almost nothing that would catch your eye. That is about to change so violently, and so fast, that it will look for all the world like a mistake in the rock. Because the somatic body, the great disposable everything else compartment, is about to stop being a mass of general-purpose helpers and start differentiating. Cells will organize into sheets. Sheets will fold into layers. Layers will become tissues, and tissues will become guts and muscles and nerves and eyes. Bodies will acquire a front and a back, a top and a bottom, a left and a right, and, having acquired a front, they will begin to put their sensory equipment there, and behind the sensors, a brain. The truce made the body possible. Now the body is going to become an animal. That is chapter 5, and it is the moment the world fills up.